Basic concepts of development. The process of development. Development is a process in which an organism undergoes a series of progressive changes, taking on the successive forms that characterize its life cycle. In its earliest stages of development, a plant or animal is called an embryo. Sometimes the embryo is contained eggshell or a uterus. An embryo does not photosynthesize or feed actively. Instead, it obtains its food from its mother directly or indirectly by way of nutrients stored in received or egg. A series of embryonic stages may precede the birth of the new independent organism. Most organisms continue to develop throughout their life cycle. Development ceases only with maturation. So, development consists of growth, differentiation, and morphogenesis. Three processes are responsible for the developmental changes an organism undergoes during its life cycle. Growth, increase in size, occurs through cell division and cell expansion. In all multicellular organisms, repeated mitotic divisions generate the multicellular body. In plants, cell expansion begins shortly after the first division of the fertilized egg. In animals, on the other hand, cell expansion is often slow to begin. The animal embryo may consist of thousands of cells before it becomes larger than the original fertilized egg. Growth continues throughout the individual's life in some species but reaches a more or less stable endpoint in others. So, differentiation is the generation of cellular specializations. That is, differentiation defines the specific structure and function of a cell. Mitosis produces daughter nuclei that are chromosomally and genetically identical to the nucleus that divides to produce them. However, the cells of a multicellular organism are obviously not all identical in structure or function. This apparent contradiction results from regulation of the expression of various parts of the genome. Once again, I'm going to read this portion of differentiation. Differentiation is the generation of cellular specializations. That is, differentiation defines the specific structure and function of a cell. Mitosis produces daughter nuclei that are chromosomally and genetically identical to the nucleus that divides to produce them. However, the cells of multicellular organism are obviously not all identical in structure or function. This apparent contradiction results from regulation of the expression of various parts of the genome. When the embryo consists of only a few cells, each cell has the potential to develop in many different ways. As development proceeds, however, the possibilities available to individual cells gradually narrow until each cell's fate is fully determined and the cell has differentiated. So, morphogenesis, literally creation of form, is the shaping of the multicellular body and its organs. So, morphogenesis results from pattern formation, the organization of differentiated tissues into specific structures. In plant development, cells are constrained by cell walls and do not move around the body. So, organized division and expansion of cells are the major processes that build the plant body. In animals, cells movements are very important in morphogenesis, and in both plants and animals, programmed cell death is essential to orderly development. Like differentiation, morphogenesis results ultimately from the regulated activities of genes and their products, as well as from the interplay of extracellular signals and their transduction in target cells. As development proceeds, cells become more and more specialized. Experiments in which specific cells of an early embryo, were marked with stains, have revealed which adult structures are derived from which parts of the embryo. The stained embryos produce what are known as fate maps. For instance, we know that the green shaded area of the frog embryo normally becomes part of skin of the tadpole larva. However, if we cut out a piece from this region and transplant it into another location on another early frog embryo, it does not become skin. The type of tissue it does become is determined by its new environment. The developmental potential of these early embryonic cells, that is, their range of possible fate, is this greater than their actual fate, which is limited to the cell type that normally develops. So, is the fate of a cell in an early embryo already determined? These cells would normally become skin tissue. Cells in this location ultimately become brain tissue. Cells here ultimately become the tissues of the notochord, a stiffening rod in the embryo. Early frog embryo with several hundred cells. Tissue destined to become part of tadpole skin is cut from an early embryo donor and transferred to another early embryo host. Tissue transplant is conducted. Experiment 1. The donor tissue is transplanted to a brain region. The donor tissue becomes brain tissue. Result. Second result, the donor tissue is transplanted to the notochord tissue. The donor tissue becomes notochord tissue, but the initial one, that is tissue destined to become a part of tadpole skin, is cut from an early embryo, that is the donor. So conclusion, cell fates in the early embryo are not determined but can change depending on the environment. And developmental potential in early frog embryo, cells that would be expected to form one kind of tissue can form completely different tissues when they are experimentally moved to other location. In this experiment, epithelial skin tissues from an early stage frog embryo was transplanted from a donor to a host embryo. The tissue that developed in the host tadpole was not skin, but was consistent with the location to which the skin graft was transplanted. Does the embryonic tissue retain its broad developmental potential? Generally speaking, the answer is no. The developmental potential of cells becomes restricted fairly early in normal development. Tissue from a later stage of frog embryo, for example, if taken from a region fated to develop into the brain, become brain tissue even if transplanted to a part of an early stage embryo destined to become another structure. The cells of the later stage embryo is thus said to be determined. Their fate has been sealed regardless of their surroundings. By contrast, the cells of the younger tissue transplant is not yet become determined. As development proceeds, cells become more and more specialized. Experiments in which specific cells of an early embryo were marked with stains have revealed which adult structures are derived from which parts of embryo. These stained embryos produce what are known as fate maps. For instance, we know that the green shaded area of a frog embryo, shown in the figure, normally becomes part of skin of a tadpole larva. However, if we cut a piece of this region and transplant it to another location on another early frog embryo, it does not become skin. The type of tissue it does become is determined by the new environment. The developmental potential of these early embryonic cells, that is, their range of possible fates, is thus greater than their actual fate, which is limited to the cell type that normally develops. Does embryonic tissue retain its broad developmental potential? Generally speaking, the answer is no. The developmental potential of cells become restricted fairly in normal development. Tissues from a later stage frog embryo, for example, if taken from a region fated to develop into the brain, it becomes brain tissue even if transplanted to a part of an early stage embryo destined to become another structure. The cells of the later stage embryo are thus said to be determined. Their fate has been sealed regardless of their surrounding. By contrast, the cells of the younger tissue transplant have not yet been determined. Then morphogenesis, literally creation of form, is the shaping of the multicellular body and its organs. So morphogenesis results from pattern formation, the organization of differentiated tissues into specific structures. In plant development, cells are constrained by cell walls and do not move around the body, or organized division and expansion of cells are the major processes that build the plant body. In animals, cell movements are very important in morphogenesis, and in both plants and animals, programmed cell death is essential to orderly development. Like differentiation, morphogenesis results ultimately from the regulated activities of genes and their products, as well as from the interplay of extracellular signals and their transduction in target cells. As development proceeds, cells become more and more specialized. So, experiments in which specific cells of an early embryo were marked with stains have revealed which adult structures are derived from which part of embryo. Next, determination, the commitment of a cell to a particular fate, is a process influenced by the action of the extracellular environment and the contents of the cell on the cell's genome. Determination is not something that is visible under the microscope. Cells do not change their appearance when they become determined. Determination is followed by differentiation, the actual changes in biochemistry, structure, and function that result in cells of different types. So, differentiation often involves a change in appearance as well as function. Determination is a commitment. The final realization of that commitment is differentiation. The role of differential gene expression in cell differentiation. Differentiated genes are recognizable by different from one another, sometimes virtually as well as in their protein products. For example, certain cells in our hair follicles continuously produce keratin, the protein that makes up hair, nails, feathers, and porcupine quills. Other cell types in the body do not produce keratin. In the hair follicular cells, the keratin encoding gene is transcribed. In most other cells in the body, that gene is not transcribed. Activation of the keratin coding gene is a key step in the differentiation of hair follicle cells. So, generalizing from examples like this one, we may say that differentiation results from differential gene expression, that is, from the differential regulation of transcription, post-transcriptional events such as mRNA splicing and translation in different cell types. Because the fertilized egg or zygote has the ability to give rise to every step of cell in the adult body, we say it is totipotent. So, because the fertilized egg or zygote has the ability to give rise to every type of cell in the adult body, we say it is totipotent. Its genome contains instructions for all of the structures and functions that will arise throughout the life cycle. Later in the development of animals, the cellular descendants of the zygote lose their totipotency and become determined. These determined cells then differentiate into specific types of specialized cells. With the differentiation, there is generally no irreversible change in the genome. Differentiation is irreversible in certain types of cells. Examples include the mammalian red blood cell, which loses its nucleus during development, and the tracheiid, a water conducting cell found in many plants. So, tracheiid development culminates in the death of the cell, leaving only the pitted cell walls that formed while the cell was alive. So, in both of these extreme cases, the irreversibility of differentiation can be explained by the absence of nucleus. So, generalizing about the reversibility of differentiation in mature cells that retain functional nuclei is more difficult. We tend to think of plant differentiation as reversible and of animal differentiation as irreversible. But this is not hard and fast rule. Why is differentiation apparently reversible in some cases, such as a plant cutting, but not in others, such as a mammalian limb? As some stages of development do changes within the nucleus permanently commit a cell to specialization. For both plants and animals, the answer appears to be no. Under the right environmental circumstances, differentiation is reversible in many cells. Then totipotency in plants, which I won't be reading because I'm focusing more on animal related aspects. Thank you. Then next topic is about totipotency. So, the ability of scientists to clone an entire carrot plant from a differentiated root cell indicate that the cell contains the entire carrot genome and that it can express the appropriate genes in the right sequence. Many types of cells from other plant species show similar behavior in the laboratory. This ability to generate a whole plant from a single cell has been invaluable in agricultural biotechnology. So, totipotency in early embryonic animal cells experiments with plants have established that somatic cells are totipotent. A more direct demonstration that all the genetic material is present in somatic cells has come from nuclear transplantation experiments. Such experiments were first done on frogs by Robert Briggs and Thomas King, who asked whether the nuclei of early frog embryos had lost the ability to do what the totipotent zygote nucleus could do. They first removed the nucleus from an unfertilized egg, thus forming an eunucleated egg. Then, with a very fine glass tube, they punctured a cell from an early embryo and drew up parts of its contents, including the nucleus, which they injected into the eunucleated cell egg. They stimulated the eggs to divide, and many went on to form embryos, tadpoles, and eventually frogs. These experiments led to two important conclusions. No information is lost from the nuclei of cells as they pass through the early stages of embryonic development. This fundamental principle of developmental biology is known as genomic equivalence, which after that we will be learning with the help of next video. Thank you.